When we think of the world's biggest problems that are yet to be solved, there's a long list. I mean, just think of it. Off the top of my head, air pollution, lack of education, poverty, and cancer. Most of these don't even have a full solution yet, and there's not one clear-cut way of approaching it. So now what if I told you that using a certain emerging technology, there's a way to cure one of these? So the focus of this video will, as you've seen from the title already, be on how we can use CRISPR-Cas9 to help cure cancer. So let's take a look at how we can leverage this to completely change the fight against this deadly disease. Starting off, we should take a look at what cancer is at a high level. Normally, when the cells in our body grow old, they become damaged and start to die. They're then replaced by new cells. Unfortunately, that's not always the case. Sometimes there are cells that just don't die, but instead multiply in number. This then causes lumps of tissue, which are known as tumors. Then in a process known as metastasis, the tumor goes to invade other cell tissues, which therefore spreads the cancer throughout the entire body. Currently, there are a few main treatment methods for cancer, but none of which are perfect. One of the most common ways is through surgery. It removes as much of the cancer as possible, but after the tumor is removed, there's no guarantee that the cancer doesn't spread again. Also, surgery can be done if cancer spread throughout the entire body, since it's not concentrated over one small area, but rather across the entire thing. One other main process is chemotherapy. Using drugs full of chemicals, chemo works throughout the entire body to kill all cells which grow rapidly. Because of this, it also kills hair cells, skin cells, and intestines, along with the cancer cells. Those side effects put your body in extreme pain, and they're a reason that hair loss and weakness are correlated directly with cancer treatment. Unfortunately, the success of chemo isn't always guaranteed. And that's the same with the things such as radiation therapy and any other treatment methods. It's now time to take a quick look at how CRISPR-Cas9 works. So starting off, bacteria captures DNA sequences from viruses inside the body, and then it uses that information to create CRISPR arrays. Using the arrays, it then remembers DNA, so it can make RNA segments if it were to come attack from the same virus again. This is the same reason people build immunity towards diseases or viruses, such as the flu or even COVID. Now the bacteria uses an enzyme called Cas9 to cut off the DNA, therefore killing the virus. In labs, this is also being used. When a guide RNA sequence attaches to a Cas9 enzyme and then a targeted part of DNA, it recognizes a genetic mutation which it's looking for. The Cas9 then cuts it off, and the cell repairs the DNA by itself, by adding or deleting parts of genetic material. Scientists are already working on using CRISPR to solve the problem. Researchers at the University of Pennsylvania are working to edit T cells, which are immune cells that can kill cancer. The therapy involved making 4 edits to the cell. One of them was adding a gene that was synthetic to give a receptor to the T cell. A receptor is similar to a claw-like protein. The receptor then finds a molecule on cancer cells known as NY-ESO1. CRISPR was then used to remove 2 genes that interfered with the receptor, so it could attach to NY-ESO1 without any issues. The final edit was the removal of a T cell gene that normally limits its ability to kill off cancer. So T cells are already a great way to combat cancer. But CRISPR levels it up in so many ways. Just right now, there's improved response of the T cells, lower toxicness, no side effects that normally come with those in current cancer treatment, and improved effectiveness. In this research, only 10% of the T cells used involved the CRISPR edits, and it still made a considerable impact on the patients tested. Two of the 4 people lost their tumors, but unfortunately they grew back. However, there still is optimism, because it's showing that there is a chance that CRISPR can be used to solve the disease. I also created a plasmid on Benchling to demonstrate this, which I'll link in the description down below, so definitely check it out. The future looks incredible when it comes to CRISPR and cancer research. It still may take time for gene editing to get out in the outside world, and there will be a few ethical dilemmas to conquer. But we might just have an upcoming cure for one of the world's biggest problems on our hands, with just a little bit more research and work. So, what's there to not be excited about? Thank you.